Okay, so we have finished primary hemostasis. We have our rather delicate, fragile little platelet plug sticking its finger in the hole of our broken blood vessel, trying to stem the flow of blood. So we're going to have to give it some help. And the way we do this is we take a series of plasma proteins, and through a series of steps, we create this fibrin mesh. And this fibrin mesh does a couple of things. It makes the blood more viscous, and it actually secures that platelet plug for us. So the first thing we need to talk about is how do we talk about, or how do we discuss these coagulation factors? As I told you in the overview, they are named by Roman numeral, and even though they're written as Roman numerals, we say them with the regular names. So this is factor 1, 2, 3, 4. We don't refer to this as factor V. This is factor 5. And the same goes for the rest of them. They all have names, and you will have to know those names. Those are common test questions, both on my tests and on the boards. And we do see many of these things referred to by name. So factor 1 is fibrinogen. We never call it factor 1. In fact, as most people probably don't remember, it is factor 1. It's almost always referred to as fibrinogen. Factor 2 is technically prothrombin. That's the inactive form. Most of the time we refer to it as the activated form, or the enzymatic form, and that is thrombin. While calcium may be coag factor 4, we don't call it that. We simply call it calcium, or calcium ions. Factor 3 is kind of more commonly, I think, called tissue factor, but it is still called factor 3. And tissue factor is the name it's given simply because this is a protein that is released by the tissues when there is damage. Factors 8 and 9, their names are useful when we're learning diseases. 8 is the anti-hemophiliac factor, and it's associated with hemophilia A. And factor 9 is a deficiency of factor 9 is the causation of Christmas disease. Factor 12 is still sometimes referred to as the Hageman factor, although that's a little bit older term. It is relatively commonly, along with factor 11, referred to as contact factor. The others are pretty much just listed by name. Sometimes 13 will be called the fibrin stabilizing factor, but most of the time I think it's 13. Now, when these proteins are just floating through the bloodstream, not doing anything, not bothering anybody, not getting in nobody's business, they are inactive enzymes or precursor enzymes. And the form, or the term for that, is zymogen. So these are zymogens as they are kind of floating through. As long as they are inactive and just hanging out doing nothing, they are zymogens, and they are known by their factor name. Once they have been cleaved, or their structure has been changed to activate them, the active form is the enzyme, and we refer to it by the number and a lowercase a. So factor 10, the active form of that is 10A, and not XA. It's written X, but it is actually the Roman numeral 10. So 10A. Calcium is not really a group. Again, just to remind you, it is sprinkled liberally throughout the entire coagulation process. I will put it into the group at the end when I put the whole mess together. PF3 is the phospholipid that we find on the surface of activated platelets, And that is also required in several of the coagulation steps. And this is what keeps the process on the surface of that platelet membrane or on the surface of the damaged cells. That's what keeps it localized and keeps you from just turning into one big massive clot throughout your whole body. There's a couple of other groups that we need to discuss. The prothrombin group, and what's kind of important about them is that they are vitamin K dependent factors. And that means that when they are being synthesized in the liver, which is where all good proteins are made, while they are being synthesized in the liver, vitamin K acts on them and adds an additional group to the end of it. If the vitamin K is not there and it does not make this change to these proteins, then they are not able to be cleaved and they are not able to do their job. So one of the ways we anticoagulate blood in the person is by interfering with vitamin K and the vitamin K dependent factors. These bind to the phospholipid membrane. So the members of the prothrombin group are 2, 5, 7, 9, and 10. And all except 9 are in the common pathway. 9 is actually in the intrinsic. And we'll talk more about that as we go along here. There are also some anticoagulant proteins that we have naturally in our system. These include protein C, protein S, and protein Z. So all of these will be interfered with if the vitamin K is not working the way it's supposed to. The contact group, these activate on contact with a negatively charged surface. They are vital to in vitro coagulation, so they're very important when we're looking at in the lab testing and what is happening in the test tube. But apparently their role in vivo hemostasis, or in keeping the blood from clotting in the body, is somewhat limited. They are involved in the process, but a deficiency of factor 12, PK or HK, don't seem to lead to a major bleeding disorder. Factor 11 does, a decrease in factor 11 does cause a limited bleeding problem, but the other ones don't seem to really have a huge in vivo role. The fibrinogen group, the most important piece about this is these are consumed as the fibrin clot is formed. So as we make this fibrin clot, most of our enzymes, most of our coagulation factors can keep working. They can keep functioning. But 1, 5, 8, and 13 actually get eaten up. They actually disappear and go away as the fibrin clot is formed. So we have to replace those if coagulation goes on for too long. Now, these coagulation proteins have 3 basic roles. They can serve as a cofactor to enable or enhance an enzymatic action. And there's a list here of which are cofactors for what. And we'll talk more about those as we go through the cascade. They can serve as a substrate for enzyme action, especially 10 and 2. And they actually can serve multiple functions pretty much simultaneously. Fibrinogen is the final substrate. Once it is cleaved, then kind of the process is over. That's the end game. So fibrinogen, once it is cleaved, it can't do much except be a meshy gel and keep things together. Enzymes, these zymogens are cleaved or they undergo some sort of structural change to become proteolytic. Factors 2, 7, 9, 10, 11, and 12 cleave various proteins at the arginine or lysine, and you won't need to remember where it cleaves them, just that it cleaves them, just that it cuts them. Factor 13A is a transglutaminase, and what it does is it breaks the fibrin residues and sort of cross-links the fibrin to make it a stronger mesh and to stabilize the clot so that it doesn't fall apart. So we're going to start with the extrinsic pathway because that's the easiest. It generally occurs within about 30 seconds of tissue damage. Factor 3 is released by the endothelial lining cells. 3 activates 7, or serves as a cofactor actually, with 7. And 7 cleaves 10 to 10A. So on the surface of the platelet membrane, in the presence of platelet factor 3, calcium and tissue factor and activated enzyme 7, they all bind together on the surface of the platelet, and it's known as the extrinsic 10-ACE because this is the enzyme, this combination is going to be the enzyme which cleaves 10 into 10A, and this is the extrinsic pathway. Okay, now we have the intrinsic pathway, and this takes 3 to 8 minutes to get going, so this isn't as quick as the tissue factor, as the extrinsic pathway. So what happens here is we have our damaged surface, and we have a negative charge on the damaged surface. So we have factor 12 along with its cofactor HK and PK. These negative ions cause these to become activated. And activated 12A along with its cofactor HK activates 11 to 11A. 11A activates 9 to 9A. And 9A hooks up with 8, its cofactor 8, to activate 10 to 10A. So on the surface of the platelet membrane, we have calcium in the presence of platelet factor 3, 8 and 9A hook up together to form the intrinsic 10-ACE. And again, this forms on the platelet surface. So here we have the common pathway. And remember, we have our intrinsic coming down here, activating 10 to 10A. And we have our extrinsic coming down this way, activating 10 to 10A. So 10A, in conjunction with 5, cleaves prothrombin into thrombin. Now, thrombin does a whole bunch of things, but the things we're looking at right now is that its primary job is to cleave fibrinogen. It is to change this protein from a soluble form to fibrin, which is insoluble, so it precipitates out of the plasma. So now we have this fibrin mesh, this insoluble fibrin. The thrombin also activates 13, and 13 basically comes in and takes this fibrin and causes it to link together, to form cross linkages between the various strands of fibrin, making this nice, tight, solid mesh. And so basically what we're looking at here is that as this activity goes on, calcium in the presence of phospholipids, PF3, with 5 and 10A become the prothrombinase complex on the surface of the membrane of the platelet. So how do we remember all of this? This is a lot of numbers and V's and I's and X's, and it gets very, very complicated. And there is a video that I'll be putting up once we get through all of these pieces that does a fairly good job with the overview. It's not real detailed, but fairly good. And I kind of stole this from him. He probably stole it from somebody else, but that's okay. It works. 10 is the key to the cascade. In coag math, it always comes back to the 10. The extrinsic pathway, 3 plus 7 equals 10. Get your calculator if you need to. For the intrinsic pathway, we just count. 8, 9, 10, 11, 12. You just have to remember that it actually occurs the other way around and you skip 10. But if you're trying to remember which ones are in the intrinsic pathway, it's 8, 9, 11, and 12. Okay, so you just count from 2 less than 10 to 2 greater than 10. But 8, 9, 11, and 12. We just skip 10. The common pathway, 5 times 2 times 1 equals 10. If this doesn't help you, ignore it and move on. But it was kind of a nice way to help remember what goes where. So we've looked at all the pieces, and when we put it all together, this is kind of what we get. This is the traditional coagulation cascade. It has changed significantly over the 30 years that I've been in the field. There was a lot of research in the 80s and early 90s into natural anticoagulation and hypercoagulable states, in other words, people who formed clots. There are a lot more feedback loops in the body when this is occurring. We have 3 basic pathways: the intrinsic, the extrinsic, and then the common. There's feedback loops all over. This combination here activates this, and this combination here also activates this. As a matter of fact, activated 11 activates itself. So there are half a dozen more feedback loops that are going on in this procoagulant cascade. Procoagulant just meaning fibrins being formed. So there's a lot more going on in here, but for evaluating in vitro testing, for evaluating laboratory tests, this is the easiest way to remember it, and this really is the most useful diagram for evaluating protimes, PTTs, and other testing that we do in the lab. I do want to point out one more time that all of this takes place on the surface of the activated platelet and on the damaged tissue. This fact that it all has to occur with that phospholipid membrane is what keeps it staying in place and not floating off to cause problems. So the results of fibrin formation: the fibrin increases viscosity of the blood, which slows the blood flow through the damaged area and reduces blood loss due to shear viscosity. It makes a fibrin mesh, which sort of tangles all around the platelet plugs, and that fibrin mesh plus the platelet plug is known as a thrombus. The fibrin also anchors the thrombus to the site of the damage to keep it from floating off and lodging in the lung, the brain, the heart, or any number of places it really shouldn't be. So that's step 2, and we'll move on to the next one. This is complicated, I know. If you have any problems, get a hold of me and we'll get it sorted.